Okay, I was having a weirdness with some Zoom, so I was afraid I would log in and nobody would be here, but it looks like we're filling in, so that's good. Good. Okay, Geffeney discussion questions, slides. Oh, I didn't prep that. That's okay. Okay, that's where we're going. This is where I want to start. Give it just another moment as people finish connecting to audio. Seeing a lot of folks just signing in. Oh, we are recording. Okay, good. Okay, let's go ahead and get started. I'd like to wrap up our sort of class discussion. Standby. To Monday. Having a little connectivity issue. There, that looks like you're on, Paul. Can you hear me now?
No. Yeah, oh, I okay. I couldn't see anything for whatever reason.
Got it. Okay, well, welcome.
I'm about done with technology for this semester.
yeah, I know the feeling.
Right. You were going to finish up?
Yes.
Right, yep. From last time?
Yep, I've just got a few slides, a couple points to make here.
Okay, before we do that, let me talk to my lab students. The technical difficulty that we had yesterday resulted in 3 of your presentations being not recorded. So I posted the self-evaluation form. I would appreciate it if you fill out that form sooner rather than later, so that you still have as much of your presentation experience present in your mind. The question and answer session for the pollen limitation group is intact, and the last presentation on the flower gold group is intact. But the first 3 presentations just didn't get recorded because of technical difficulties I was having, because evidently here in the last week of class, Zoom hates me. So my apologies for that. I tried to recover it a couple of different ways and just couldn't get access to the files. So the form is on Moodle. Fill it out as soon as possible, but at the latest by Friday. I realize I told you in class by Monday. The Thursday group, Dr. Judd will tell you what the timeline is for when he wants that done for the Thursday group. Shouldn't take you too long, but once again, do it while the experience is still fresh in your mind. This is one of the few assignments where you're welcome to talk to other members of your group as well, if you need to remind yourself kind of how things went and things like that, especially since you don't have a video to remind yourself of what was happening. All right, that's it. Thanks, Nathan.
Okay. Yeah, look for an email from me this afternoon regarding our plans for lab. Okay. Share screen. So hopefully you all saw you were able to follow the discussion of the residuals that Dr. Polinsky shared with us last time, and how the authors adjusted for both mass and speed in the snakes in order to compare resistance among the different groups and among the different populations of snakes. Slide. Okay, so this is a slide that I wanted to make sure we talked through. Constraints and trade-offs are intimately related. So what's being shown here is the relationship between baseline speed and post-injection speed. At the end of class last time, I asked somebody to tell me what the dotted line means in each of these graphs. Can someone remind us what the dotted line represents?
Isn't it just one?
What's one? What about the line is one?
Is like what they predicted?
You're circling the ideas, but put it together into a complete sentence. So the line has parameters, right? Which parameter is one?
The slope.
Right, the slope is one. So the line shows the predicted post-injection speed based on the baseline speed if there's no effect, or no trade-off, excuse me, no trade-off. That's why if you go, for example, in this Benton graph, right, 15, go up to the dotted line on the, go up to the dotted line from 15 on the baseline speed, and then go over to the y-axis, and you're down here around 8 or 9 on the post-injection speed. That's just the effect of being injected if there was no effect of tetrodotoxin. Then the solid line, by contrast, how is the solid line different from the dashed line? Mathematically or graphically, how is it different?
The slope isn't as steep.
Right, the slope is shallower, and this is in fact the best fit. This is the regression line through this cloud of points, through each of these clouds of points. Now, their statistical investigation was, is this decrease in slope, or is the difference between a slope of 1 and whatever this slope is that's less than 1, is that difference statistically significant or not? What's nice about showing all these together, at least these three on the left together, is all three show a change in the same direction, right? All three of them show the shallower slope than the 1-to-1 line. What does a shallower slope mean? If the solid line really is significantly shallower than the dotted line, what's the interpretation of that?
That after the injection, it has more of an effect on their speed.
Can you repeat that? I just happened to miss it.
That after the injection, there was more of an effect on the speed.
In which snakes? In the case of the dotted line, there's an effect on speed no matter what kind of snake you are, whether you're a fast snake or a slow snake. But in the case of the solid line, let me see if I can have another way of asking this. How does the effect of injecting TTX affect slow snakes relative to fast snakes?
Slow snakes have more resistance.
Right. The slowest snakes, the snakes with the slowest baseline speed, have the most resistance. That's what's shown by this shallower slope. That deviation from a slope of 1 is depicting the trade-off. The reason that there's 2 columns of graphs here is because these points on the left are not independent observations in a strict statistical sense. Why might that be, right? What do you know about the snakes they used, how they organized them, how they sampled them, that would make it so that an observation of resistance in one snake is not statistically independent from the observation of resistance in or change in speed in another snake. In what ways are the snakes similar to each other?
A lot of them are related, aren't they?
Yeah, they're siblings. Good. Um, so that means that they have similar genetics. That means they have similar environments during development. That means that they've had a lot of things that are sort of in common. And we want to remove some of those effects. So on the right side, what you're seeing is the same analysis, but this time it's the family means. Each point is the average baseline speed and post-injection speed of a group of siblings. So when we started discussing this graph, I said that trade-offs and constraints are intimately related. Let's look at this table. And we're going to look at the right side, the family means, where we go for kind of a more fair comparison, because we're not sort of pseudo-replicating observations within families. What's different about the slope parameter, B, in the mass-adjusted Bear Lake regression versus the mass-adjusted 10 Mile and Benton regressions on family means? So the parameter B represents the slope of the regression. And once again, the asterisks down here tell you about the significance levels for these comparisons. And we're using, on this table, they use the T statistic to evaluate the regressions. I think I'm asking the question in a way that's a little bit challenging. Let me give you the idea at the beginning, and then I'll ask again. It's the costs associated with resistance that that set up a trade-off. So you have to, in order for a trade-off to appear and be evident, you would expect that there's got to be some advantage to TTX resistance. Why would there not be an advantage to TTX resistance among the Bear Lake snakes?
They don't eat the newts, so there's no reason for them to have the resistance.
Right. We might expect there's some variation in their resistance in general, because that variation had to be present for the arms race to get started. But notice the relationship is much weaker, and if you don't adjust for mass, if you use the unadjusted regression, the relationship the regression between baseline speed and post-injection speed is not significant here. That's probably because there's no fitness advantage to being resistant if you're not eating TTX prey, if you're not eating newts. Okay, the last two questions in our discussions list was, what was their hypothesis for the explanation of resistance in these snakes? Who caught that? Why do they think, what things do they think besides mass and speed might contribute to this genetic basis of TTX resistance? See who's all signed on here. Did you all identify what mechanism, what sort of physiological or molecular mechanism they suggested could explain this trade-off?
Is it differences in their sodium channels?
Good. Yeah. So they hypothesized that differences in the function of the sodium channels might might contribute to blocking tetrodotoxin. Um, or sorry, they suspected that changes in the function of the sodium channel affect perhaps the binding of tetrodotoxin to the sodium channel, such that if you've got a sodium channel that is not as affected by TTX, isn't blocked by TTX, it also maybe has kind of lower conductivity or slower kinetics of how the sodium channel allows sodium ions to move through it, thus affecting muscle performance and therefore speed in this case. They use speed as their bioassay of muscle performance. Okay, and then the last question was, let's see, I've got it right here. Too many windows. Why are trade-offs necessary for a predator-prey arms race to be sustained? This is asking sort of what are the attributes of a coevolutionary interaction that make it an arms race, and what about the trade-off keeps something in this arms race scenario, keeps an interaction in this arms race scenario. Let's go around and I want to ask, what are some attributes, what makes a coevolutionary interaction an arms race? Let's see, Daniel, can you give me an attribute?
When there's like a predator-prey interaction and the predator has a defense that the prey, the prey has a defense that the predator has yet to fully adapt to.
Okay, good. Let's keep going with that. I want to build on it. Who wants to add to that?
So once the prey like has the attribute, the predator tries to like adapt to that attribute, and so then the prey will adapt even more.
Good. You can avoid that sort of anthropomorphizing by remembering that you have a fitness advantage if you can overcome the defense of the predator, instead of saying that the predator is trying to defend it or trying to overcome that defense. But good, that sort of reciprocal increasing is what matters. And now, so let's add to that now the trade-off. What's important about the trade-off that would help keep that the evolution of a defense and overcoming the defense in kind of a lockstep, roughly matched level? Why not just become super resistant to TTX and take over the world as a garter snake?
Because then they might like cause the food source to go extinct.
That's not where I'm going. Let's see, how do we say this? Let's say you do drive the newts extinct. Let's take it to the thought experiment extreme. The newts go extinct, the snakes get really, really hungry, so eventually they start eating alternative prey. Would they stay super resistant, or would there be a cost associated with remaining super resistant?
Well, since they became like super resistant, wouldn't they, I don't know, become slower, I guess?
Yeah, if they evolve super resistance, you'd expect them to be really slow. If they're really slow, they're vulnerable to predation by birds or cats or foxes or whatever. So it's the cost of resistance that keeps the snake's ability to overcome the newt's toxicity matched to the newt's toxicity level in each population, in each sort of tile of the geographic mosaic. Does that make sense?
But what if we found out that like they kept getting super resistant and they weren't getting slower? What would like happen then?
If there's no cost to being more resistant, then you wouldn't expect there to be an arms race. You would just have a predator that's overcome the defense of the prey. We're actually going to get to something really similar to that today or Friday. We'll be talking about that kind of a thing and how that might actually happen under certain conditions in a predator-prey arms race like this one. So that's a good question. That'll lead us right to it. Okay. Oh, I didn't realize I was still sharing. I was over here. Okay, that's it for the Brody and Brody 1999 paper. Let's see. I'm just double checking to make sure that you guys have the discussion questions for Geffeney posted.
I loaded them already.
Okay.
Earlier today.
Thank you. Okay, so you should all have the discussion questions. We have a few slides to talk about as we come back together as a class. And
and I want to give a little bit of an introduction before they go away.
Yeah.
So while I'm doing that, if you could set up the breakout rooms.
Yep.
You're the host, as it turns out. All right. Can you guys see my screen?
Yes, sir.
All right. Okay. So the Geffeney paper from 2005 in Nature, basically it's dealing with the resistant snake that is resistant to tetrodotoxin produced by the toxic newt. And the idea here is that sodium channels are actually what is being influenced by tetrodotoxin. So whenever you have a nerve impulse generated, it's due to the influx of sodium from outside the nerve to inside the nerve, which changes the charge on the nerve membrane from a net negative charge to a net positive charge. And then as it pumps sodium out while pumping potassium in, it reestablishes that negative charge of more sodium ions outside the neuron than inside the neuron. And when the sodium channels open, sodium flows into the neuron down that concentration gradient. This is kind of molecular biology models of what sodium channels look like, and the voltage-gated sodium channels that you guys are dealing with look like this, and they have 4 domains. This is domain 1, domain 2, domain 3, and domain 4, and each of these domains makes up a separate unit of this thing that kind of makes a hole through the membrane, which allows the sodium channels to pass through from the outside of the membrane to the inside of the nerve. This is just a blow up of it. Once again, this is domain 1, domain 2, domain 3, and domain 4. And the other thing that you might have had some difficulties with is this idea of patch clamping. I put a link to a YouTube video illustrating what patch clamping does. But what patch clamping does is they use a really tiny pipette to basically suck the cell membrane of a nerve up against the pipette. And the pipette is filled with electrically conducting fluid. And underneath this pipette in the membrane are sodium channels. And then when those sodium channels open, it changes the charge of the liquid that is in here, and there's an electrode, because as sodium channels move out of the electrode into the cell, in this case, rather than the other way around, that change in charge is registered by a little tiny electrode, which is also inside of that pipette. And if the sodium channels are blocked by tetrodotoxin, then that charge won't happen. And so this is actually a picture of what this little tiny pipette looks like touching up against a nerve cell body here. So this is an actual image from a hippocampal neuron. And this electrode will then measure any change in the membrane potential on the membrane of this nerve. And so when they talk about patch clamping and how they measure the depolarization, this is the method that they use. And I think with that, we can go to the breakout rooms. And Dr. Judd, I'll just text you when I want to change rooms. Any questions about that information I just gave you?
I was having a little bit of trouble with getting these set up, so they're almost done now.
Okay. What room are you working on?
I'm working on 2 now.
Riley, Grant, Keaton, and Paula.
Yes.
And number 3, I've got
3 is Casey, Dominic, Kat, and Cameron.
4 is Tessa.
Elena, Sophia, and Connor and Ryan for 4.
Okay.
Room 5 is Angelina, Hunter, Sydney,
Sydney.
and Katie. Room 6 is Daniel, Alexis, Michaela, and Devin's no longer with us.
Okay. Two. Okay, they should be open. And we're good.
Great, Kat's still around.
I have 0 unassigned. If you don't, Kat, if you're not seeing the window, just see if it's maybe behind your Zoom window or something like that. There you go.
All right, so the first question is asking if they made the point that regulations of genes of major effects rather than the alteration of functional products might explain much phenotypic diversity. What does this mean, and why are they drawing this distinction in the introduction?
This is an important idea in biology right now, especially in the early 2000s when it was really getting going. So in order to make it a little bit more concrete, ask yourself about the sodium channels that they discussed that are present in very, very young snakes and how those differ from the sodium channels that are expressed in mature snakes. That was this paper, right? They talk about young snakes. What do you guys think they mean by regulation of genes? What do you think that means?
Well, I know that they talked about like how the genes were actually developed or something like that.
Whenever I read the regulation of genes, I almost thought of like the fitness landscape. In that, like, you know, it's proactively evolving to you know, adapt better to its environment.
When we talk about gene regulations, I think both of you have some imagery in your head that's relevant, but let me help try to clarify some things. Almost every cell in your body, and the same is true for the snakes, has a complete genome, right? So you have all of the genes are present. But are all of your genes expressed everywhere in your body all the time? The genes that activate other genes that result in the development of an eyeball are only active for a little while, very, very early in development, and then they're turned off. And then you end up with two eyes right here instead of eyes all over your body all the time, right? So that genes vary in where they're expressed and in how much functional product, how much protein or enzyme or whatever else, or RNA is produced. So regulation refers to where and how much of a gene's product is produced. They mention later on in the discussion that the very youngest snakes have sodium channels that are resistant to TTX. But then once you're an adult and you're hunting, then you've got these sodium channels that are vulnerable to TTX. So if there was a change in when those juvenile sodium channels are expressed, how could that result in resistant adults? A good
So
analogy.
like if it
Go
doesn't
ahead.
go away after they're like as they're growing up, they will just keep that resistance.
Yeah. Yep. So that was one hypothesis that they had to be aware of. And that's why they talk about regulation of genes, because there's not a new gene that evolved to give resistance. It's just that one, they change. There could have been a change in the timing of when this other gene is expressed. That might sound familiar to you if you know about lactose intolerance and how the incidence of lactose intolerance is much lower in people of European descent and North Asian descent, because the gene that breaks, the protein, the enzyme that breaks down lactose is expressed in adults in people of European descent. And it's generally not expressed in adults from people from elsewhere in the world, say from the Americas or from the African continent, something like that. Anybody want to expand on that or ask a question about that before you go on to number 2?
Where was it in the paper that they talked about like the young having that resistant?
I was just reviewing this right before lunch, and I'm pretty sure that it is in the, let's see here. I thought I had it highlighted, but I guess I didn't. No, I will give you an answer in just a moment. In the meantime, how about the goals of the paper?
Well, I guess like the goal was kind of to just figure out why the snakes were resistant.
Good. Yep, that's correct. You can make it. You guys can also kind of build on it and make it more specific. So I have the same thing written down here, but also with kind of more specific versions of that. So once they got it to the sodium channel, in what way did they get more specific than that for figuring out what gave the resistance?
Well, they went and tried to see, like, I think they were genes, and see like whether it was that 1.5 or 1.
Yeah, so the TSNAV 1.4 is a gene. The TS stands for Thamnophis sertalis. NAV stands for voltage-gated sodium channel. And then there are several versions of that gene, like 1.4 and 1.5. And I think we're going to talk about that as a class in a little bit. I need to check on another group because we're going to come back together a little sooner than we have. But see if you can get through at least through 2 and 3 before we come back together as a class. And I will I'll put the information about the juvenile version of the gene in the chat, or we'll talk about it as the class because I gotta make sure I hit everybody. Sorry, I thought I had that highlighted.
Hey, Judd. We are confused.
Where are you? Tell me about it.
Well, so we weren't really able to answer the first question very well.
What do you guys think gene regulation means? What does that mean to you?
Like monitoring different genes in a DNA sequence, or
let me ask another question that'll make the answer a little more obvious. Except for your red blood cells, every gene in your entire genome is present in the nucleus of every cell in your body. Are all of your genes expressed everywhere all the time?
oh, no.
No.
That's what regulation is. It controls the timing and the amount of gene expression.
Okay.
So with that understanding, what does question 1 sound like now? Does that sound a little more easy to answer?
So I think that maybe something that was kind of asking was that you would have to have experience in each class to be able to understand exactly how action helps you.
While you're thinking, did anybody find a spot in the paper where they talked about sodium channels in baby snakes, in really, really young snakes? I'm looking for that part of the paper and they didn't highlight it.
I'm not sure where the baby snakes thing is, but would the first question be talking about more so how like genes not being expressed changes phenotypic diversity more so than like altering one specific gene? Like,
It can, yep. That's the idea, is that changing the amount or the timing of a gene's expression can change the phenotype of an organism, just like changing the sequence of the gene could affect the phenotype of the organism.
okay.
Okay, good. Take a stab at number 2 and being as general and then again as specific as you can. Take sort of a really general and a really precise way of answering what's the goal of this paper. And I'm going to check on one more group. Actually, I got to try and check on 2 before we go back together as a class.
Okay, so the goal of this paper would be more so to look at how the genes were different in the expression of like sodium channels, or not sodium channels.
No, you're good. So if gene regulation is not the issue, and it is in fact different mutations in the genes, then the rest of the paper follows, right? Then they would compare the sequences of the snakes in the different populations, right?
Mm-hmm.
So go with that. I'm gonna go here. Hello. I think you all might be the only complete group. Like all of you are here. I'm just gonna jump right into it because I wanna make sure I check on each of the groups. What do you guys think it means when they talk about gene regulation? What does gene regulation mean to you for the first question?
Okay, so that was kind of an area where we were kind of confused. But we thought it was like the like genes, whether they're like, I don't want to say like on, but like functioning or versus not functioning.
You can say on, people say that all the time. The word you're looking for is expressed, or the genes being expressed. But people talk about genes being on and off all the time. Good, so aside from your red blood cells, every cell in your body has your complete genome in it. Are all of your genes expressed in all of your cells all the time?
No.
That's what gene regulation is. It's control of the timing and the level of expression of your genes. So if gene expression is not what's going on here, if gene regulation is not what's going on, then what is going on is mutations in a gene, changes in what the product of the gene is, not in how much is made or where it's made or when it's made. The rest of the paper follows from that hypothesis, that there are mutations that are important for resistance. They've then acknowledged that gene expression could also play a role, but that's not the problem they're going to try to tackle here. Does that make sense? I'm trying to find something about that while you guys are talking in the paper that I forgot to highlight. So I'm going to keep doing that. In the meantime, what did you all come up with for the goal of the paper?
Well, we kind of said like how the toxins affect like the DNA, well, like the sodium channels within the muscle skeleton. So how the sodium or how the toxins kind of affect it.
Good. And why might the toxins affect the sodium channels differently according to them?
Is this where like the DNA comes into play?
Mm-hmm.
Okay,
Yep.
yeah. So just like the difference in their DNAs and how they each react differently, I guess.
And so what is a DNA sequence translated into? Going all the way back to when we were doing DNA and central dogma stuff.
Amino acids.
Good. Yeah. So amino acids. So it's not, in fact, the DNA sequence itself that the TTX interacts with. It's the amino acids that have been folded together to make the sodium channel protein itself. Right? And so those amino acids have molecular structure. And they looked at how the different sodium channels vary among the different populations and what region of the sodium channels had mutations that seemed to matter. Which region seemed to matter?
Was it domain 4, I think?
Mm-hmm. Yep, and they talk about the pore helix and the beta strand especially, which we'll talk about as a class here in about 3 minutes. Yeah, so they wanted to identify how do these sodium channels vary and what parts of the sodium channel specifically varies in a way that matters for TTX toxicity. Good, I'm gonna check out one more group, so keep going with the last few minutes here and then we'll come back together in just a couple moments. Breakout room. I see.
We had a question about 2C, why did they begin with transcribed RNA?
Oh yeah. What does transcribed RNA get translated into?
Proteins and amino acids and stuff.
Right, yep. So sometimes there are segments in a gene that, in the DNA, that don't get transcribed into RNA and therefore never become part of the amino acid sequence. So it's not worth looking at those if what matters is the amino acid sequence of the protein itself. Does that help?
Yeah. And then also we kind of struggled with number 4 as well.
Good. Oh yeah, okay, this is one we're gonna talk about as a class here in about 2 minutes, because it's 1:19 already. But remember that at this scale, you're dealing with atoms and molecules. So the TTX has a structure, and around that structure is the electron clouds. And so there are charges, positive and negative charges at different ends of the TTX. And it's also got a specific shape. So when that molecule interacts with the sodium channel, not only does it kind of fit or not fit, depending on resistance, into the channel opening, into the pore, but also the charges that are around the sort of the outside of the molecule interact with the charges that are on the surfaces of all of those amino acids. So that kind of physical relationship, that physical molecular relationship is kind of closely related to the proposal that they actually give. And it's 1:20 now, so I would keep going, but Dr. Quincy is going to pull us together for the whole class here in just a second.
Okay, thank you.
Mm-hmm.
Thank you.
Okay. I got a message that maybe my my screen wasn't sharing earlier. Is that true?
Well, it was sharing, but it wasn't like going through the slides, I think, which I
Okay. All right. Can you see my slides now?
just,
Are they changing now?
oh, yeah.
Okay. Snake newt. Action potential. Sodium flows into the nerve, changing the polarity on the nerve membrane, and then the action potential is restored to a resting potential until the next nerve impulse comes down. This is accomplished by the sodium channels. This is the sodium channel that we're interested in here. And yeah, I just really don't like some things about Zoom. So this is the sodium channel. It's made up of these four domains: domain 1, domain 2, domain 3, and domain 4. It basically forms a pore that goes through the membrane of the cell. This is what it looks like blown up. You can see the four domains. You still getting the new slides?
Yep.
This is what patch clamping is like. It's got a pipette pressed up against the membrane. Underneath this pipette are sodium channels, and as sodium ions flow into and out of the cell through the sodium channels, it registers a current in a little tiny electrode that is stuck in the fluid inside of this pipette. And this is what it actually looks like. You're looking through a microscope at the pipette, pressing against the membrane of an individual nerve cell body. And anytime that this nerve cell body changes polarity due to the opening, and for that matter, closing of sodium channels, it will register a change in current on the electrode that is inside this really, really tiny pipette. And that's actually how they measure changes in the membrane, the charge on the membrane in these cells. So to kind of work through the questions, because I think in the participants tab, register for me, raise your hand basically if you found this to be a little more difficult of a paper to follow than the previous papers. Okay, I see some hands up in videos and I see some hands up. What was difficult about it?
There was just a lot of terms I was not sure what they meant.
Okay, did you go and look them up, Keaton?
Oh, it was me, Ryan.
Oh, that's Ryan. Oh, sorry.
Yeah,
did you go?
I didn't look up all of them, but yeah.
Okay, but you had to go look up some things because it's all new to you. Anybody else? What made this particularly challenging?
It was just really dense with information.
It is pretty dense. That's the nature. That's kind of the nature of nature and science papers. You notice that they change the order of things. The methods come at the end, and the methods are in really tiny type because they're trying to save as much space as they can. So the structure of the paper is a little bit different. How many of you, be honest now, how many of you didn't read the methods section that was in a little typescript at the end?
I thought it was its sources.
Oh no, all that little type at the end is methods, and it's important for you to actually go and read those because there's good important information in there. Other challenges for this paper for you? What were some other challenges you found?
I feel like I couldn't imagine what was happening. Like with the other papers, I knew, like I could imagine, oh, they're racing newts. I know what that means. I know what that's, or snakes. I know what that's going to look like. But with this, it was kind of like really hard to conceptualize.
Okay, really abstract. Okay, so let's start going through the questions and we'll try and get through as many of them as we can. So they looked at 4 different populations. Bear Lake, which they looked at previously, and actually I think I have a location wrong. I think it's further down in Idaho, actually. This is from the next paper that you're going to read, and it shows a bunch of populations that they've looked at. But in this paper, they looked at Bear Lake and Warrenton and Benton County and Willow Creek. What's the difference between these 4 populations? Why would they choose these 4 populations? The answer is in figure 1.
They're different populations with different resistances.
Yeah, who was that?
Katie.
Okay, yeah. So if you look at this, 3.6 mass adjusted mouse units, 15.2, 34.1, and 730. This is the amount of toxin that they had to give the snakes in order to reduce their locomotor speed by 50%. And so you can see it takes a lot more tetrodotoxin to slow down a Willow Creek animal than it does to slow down a Bear Lake animal. So we have non-resistant, a little resistant, more resistant, and super resistant snakes in this population, or in this sampling scheme. What is the section on the right-hand side of this figure doing, showing you?
Is it like differences in their DNA?
It's not differences in their DNA. What are those? Those letters aren't nucleotides. What are those letters referring to? This I stands for isoleucine, and this L stands for leucine. This I stands for isoleucine, this V stands for valine.
I know one of the groups I talked to explained what these are.
Mm-hmm.
Are there different amino acids?
There are different amino acids. And so they reflect differences in the DNA, but they're differences in the amino acids. And you can see that Willow Creek has a bunch of different mutations, this leucine, this valine, this asparagine, and this valine. But the only place where they're really different from any of the other populations is at this residue that's a leucine and these 2 residues that's an asparagine and a valine. And so these 3 things might be the ones that are responsible for making Willow Creek really, really resistant. Although if you add a valine here at 1561, it makes you somewhat resistant.
Just to be clear, those dots mean that the position for that, the letter at that amino acid position is the same as on the top row.
Yeah, yeah. So all of these letters, all of these amino acids are the same here and here and here, and they're just showing you the changes. Yeah, good point. So they figured all of this out by basically getting messenger RNA and converting it into what's called cDNA. Who can tell me what messenger RNA is relative to cDNA? How do those 2 things differ? And how do you get cDNA out of messenger RNA? Ryan, this is why we read the method sections. Think back to central dogma. What does central dogma say happens in a cell when you're expressing a gene?
Casey or Kat, I know you 2 were real close to this one when we came back together as a class. Can you tell us what did they make the cDNA out of, and why they did that?
I went through this with one group also, group 5.
So they like took the cDNA, or to make the cDNA, they like took the mRNA that had been transcribed from the DNA and then like basically transcribed it back into the DNA, but it was cDNA, so it was like had kind of the different properties that came from the mRNA that would have gone to the protein instead of having all of the different stuff that comes with the DNA itself.
Okay, so when you have a gene might be of a particular length. And then that gets transcribed, that length of DNA gets transcribed into a length of messenger RNA. But after the messenger RNA is built, it actually goes through what we call post-transcriptional editing or post-transcriptional processing, and chunks of that messenger RNA get cut out. And so the messenger RNA that gets shipped out into the cytoplasm is oftentimes smaller than the messenger RNA that came from the DNA. So DNA to original messenger RNA, and then post-transcriptional processing shortens that so that the messenger RNA that goes to the cytoplasm is the messenger RNA that actually gets transcribed into a protein. And so by starting with the messenger RNA that's in the cytoplasm, you know that that sequence of messenger RNA is actually going to get translated into a protein. So by taking that messenger RNA, using reverse transcriptase, which takes messenger RNA and converts it back into cDNA, you basically are just getting a DNA copy of the actual coding sequence for that gene, not a bunch of the junk DNA that gets excised out of that gene during this post-transcriptional processing. And so that ends up being a much smaller section of DNA, and it's the DNA that is really responsible for, you know, what was this 1,585 amino acids that make up this this amino acid, or this protein rather, the sodium channel. Then they took that sodium channel that they had created, the DNA for that sodium channel that they had created, and they put it into frog eggs. Why would you want to put snake DNA into frog eggs? Especially xenopus eggs.
Shall we make preparing an answer to that question homework for start of class on
We
Friday?
should. Yeah. So do a couple of things for me between now and Friday. Look up xenopus. It's a type of frog. See if you can get a picture of xenopus with eggs, so you have some idea of the scale of a xenopus egg. The other thing that I want you to do is I want you to look up what a chimera is, so that you know what chimeric DNA is. And then ask yourself, why would they take chimeric DNA and pay attention to what parts of the chimeric DNA were from one species and another species? Why would they put that in an egg? What does that gain the researchers in terms of figuring out whether or not sodium channels from different types of snakes, different populations of snakes, actually are the thing that is imparting resistance to these snakes? These points, the discussion questions that deal with, well, let me see, let me pull up the discussion questions. They're hidden behind Zoom. Questions D, E, F, G, and H, those are all really crucial for you to understand what's going on in this paper. And between now and Friday, if you have questions about this, email Dr. Judd or I. It's crucial that you understand what's going on in this paper, because these same methods that they use for measuring resistance in sodium channels, they use in the paper that we're going to be discussing on Friday. So if you don't have a good handle on this paper, you're going to be really lost come Friday.
So go over, yeah, so look up xenopus eggs and what a chimera is. Read carefully on page 760, and if you didn't do the methods, see if you can make it through the methods.
Yeah, definitely hit the methods if you didn't hit the methods. I think that's going to help you understand some of these things that they're talking about. It's in really tiny script. Break out a magnifying glass if you have to. All right, we'll see you guys on Friday, if not before.